unit, whereby we will discuss electrophoresis and affinity chromatography. So just like yesterday, in case you have a question, just unmute yourself and speak out. I will not be able to monitor the chat. So as an introduction, you just describe what electrophoresis is, and that will be our first activity, electrophoresis. So this is a separation technique which is based on differential movement of charged solids under the influence of applied electrical fields. So this is something that you have done or heard before. It's not any different. So the separation will be influenced by a number of factors. And one of the key factors is the charge on molecules. And as we have seen in our series of discussions, the pH and the pKa is critical. But in electrophoresis, we can also separation can also be achieved by utilizing induced charges. So you may have some neutral molecules being attached to a charged group and thus facilitating separation. For example, barium associates with the neutral sugars and can therefore be used to carry out electrophoresis on sugars which are neutral. We may also consider iso or amphoteric substances whose net charge depend on pKa of the different groups. So depending on the pH of the media, the group or the molecule may be either positively charged or negatively charged, or have a net zero charge, which is the isoelectric point. So for the weakly acidic or weakly basic, the degree of ionization and hence the mobility during electrophoresis will depend on the proportion of the ionized function. So, or the ionized species. So if a higher percentage is ionized, depending on the pH, then the movement through the system for positively charged, and we'll explain why positively charged, will migrate faster. So types of electrophoresis. We have two main classifications or two main groups, and these are moving boundary electrophoresis and zone electrophoresis. There are subtypes under each of them. Under moving boundary, moving boundary electrophoresis, the migration is through the migration of ions is through a solution. And this type of electrophoresis provides information on mobility of the solute and isoelectric points of analytes. The subtypes include capillary electrophoresis, isotachophoresis, isoelectric focusing, and immunoelectrophoresis. Under the zone electrophoresis, the migration of charged molecule is through inert and homogeneous supporting material. These materials may include paper, gel, or cellulose membrane. So the subtypes of zone electrophoresis will be named according to the supporting medium. So we have paper, cellulose acetate membrane, gel electrophoresis, and under gel we have agarose or polyacrylamide. So what is the basis of separation in electrophoresis? Separation implies that, in chromatography, we always talk about the differential movement. So the ions exhibit differential velocities. So the rate of movement of each ion is influenced by certain factors. And that allows for their separation in terms of space. So these factors, or the factors that have a role to play in electrophoresis, include electric field. So this is the voltage applied across the separation medium, the charge of the ion. So you may have singly charged ions, multiply charged ions, 2, 3 positive charges, etc. So that the amount of charge on each ion will influence that. Then we have the mass of the ion. So this is obvious because the force experienced by similarly charged molecules will be the same. Therefore, the rate of acceleration and migration will be influenced by the mass of the ion. The other one is shape, and shape and mass will influence the resistance of the material. So whether it's a gel or a solution, the resistance that the ion will experience will depend on shape. Okay. So rate of movement or the velocity of the ion which we can use mu seems good. So the rate of movement as a function of the charge or the power applied to the separation medium is called electrophoretic mobility. So mu EP will be given by the velocity you divide by the applied voltage. So E is the applied voltage, V is the velocity. So that ratio gives us what is known as electrophoretic mobility. So this has been observed to be the same as, or it is dependent on the charge on the ion, which is Q, divided by the resistance to movement, F, or what F actually represents frictional coefficient. Okay. So these terms, or the frictional coefficient, depends on the Stokes, what is known as the Stokes radius. So Q divided by 6 pi NR. So Q, same charge of the ion, pi just the normal value of pi. This represents the viscosity, while R is what we are calling the Stokes radius. Okay. So electrophoretic mobility will therefore depend on the viscosity, the radius, the charge of the ion, and applied electrical field. During electrophoresis, the applied voltage is usually constant, and therefore the movement of the different ions will be influenced by the mass and shape, as well as the charge on the ion. So if you have molecules, two molecules which have the same shape, maybe they are spherical. If they are spherical, then the influence of shape is minimal. So you can say that the electrophoretic mobility of the two ions will only be influenced by the mass and the charge. So you can actually plot, you can have a plot of mass to charge ratio depending on the retention time through the system. Any questions on that? So our two interests of electrophoresis, we only discuss two types, which are page type of zone electrophoresis and capillary zone electrophoresis. So those are the two that we will look in detail for now. So as we mentioned when we were discussing the types, we say gel electrophoresis comprises of several types depending on the medium that is used for separation. So our main interest is the page polyacrylamide gel electrophoresis. Separation in page is achieved through molecular sieving. So this gel has small pores through which molecules will be migrating. And because of the interaction of the pores and the molecular size, sort of sieving is therefore achieved. Gel, we can say in terms of nature, is a colloid made of homogeneous non-crystalline material dispersed in water. So in this polyacrylamide gel, it's just small particles of polyacrylamide dispersed in the water. It is electrically neutral. In this particular case. So the other types of gels are listed there. So this polyacrylamide is prepared by polymerizing acrylamide monomers in presence of methylene bisacrylamide. So this is acrylamide and this is bisacrylamide, methylene bisacrylamide. So the second one, the second molecule allows for cross-linking, and therefore the proportion of it will determine the pore size. Polyacrylamide gel is thermostable, is transparent, is strong, relatively chemically inert, neutral, and the pore sizes will be determined by the proportions of the two monomers. It offers certain advantages compared to the natural gels, which is the agarose or cellulose. One of them is that it has high resolving power for small and moderately sized proteins and nucleic acids. The other advantage is that it can hold large samples. Okay? So it can be used for sort of purification. You can get pure samples or pure analytes which can then be used for other purposes. The third advantage is that there is minimal interaction between the migrating molecules and the matrix. And fourth is that it has physical stability compared to the other gels, especially when you consider pH and temperature. However, it also has certain disadvantages. One of them is that the monomers, acrylamide, is a neurotoxin, so it's a toxic material. If it comes into contact or is ingested, then it can cause toxicity. And it's also suspected to cause cancer, so it's carcinogenic. So it requires careful and special handling techniques. In addition, some other reagents which are required for the formation of this polymer also require special handling techniques because they are unstable. And third, making gels of reproducible thickness and composition is difficult, and therefore results may not be reproducible over long term or during the different separations. So reproducibility of separations poor. So those are the three advantages. The monomers are neurotoxic. They are also carcinogenic. Catalysts are unstable, require special handling techniques, and the reproducibility of results or separations may be difficult because of the difficulties of producing similar or gels with similar characteristics. So page can be classified into different types based on separation conditions. So there is what is known as native page. And in native page, the analytes are, and in this case, we are considering proteins. The conditions are non-denaturing, so they don't destroy the structure, especially the tertiary structure and quaternary structure of the proteins. So these proteins would be expected to exhibit biological activity. In native page, separation is based on the charge, size, and shape, and can be used for purification or separation of proteins in their original or natural characteristics. Then we have denatured page, also known as SDS page. So SDS represents sodium rhodesyl or lauryl sulfate, which is a surfactant. So this sodium rhodesyl sulfate, a soap, destroys the quaternary structure or tertiary structure of the proteins, and therefore separation in denatured page is based on molecular weight of the proteins. SDS being a soap has two components which are oppositely charged. Okay, you know the structure of a soap. So the SDS masks the charge and confers a net negative charge on the protein, which is proportional to the length of the protein. So the protein SDS complex is negatively charged, and the total charge depends on the total length of the protein. In some cases, heating the protein with mercaptoethanol helps to break down disulfide linkages. So if complete denaturation is required, then heating with mercaptoethanol breaks these disulfide linkages and therefore completely denatures the protein. So this SDS page is used to determine molecular weight of proteins, and it is also useful for checking the purity of proteins. SDS page can be performed in two formats. Can be either horizontal or vertical. A vertical is also called column page. So the first depiction there is a column page whereby the gel is held in a column and is inserted between two separate buffer reservoirs. The upper reservoir contains the cathode and the lower one contains the anode. And in this column page, electrophoresis is carried out in basic pH, under which conditions most biological polymers, the proteins, will be anionic. So they will be moving towards the anode. The sample to be analyzed is usually placed on top, on the top of the gel, and voltage applied. A tracking dye is also applied, and this dye would move faster than any of the components of the mixture, and it is used to indicate the completion of the process. So it would reach the end of the column before any of the components. Therefore, any other component in the dye will be behind it. So once the dye indicates that the end has been reached, or it has reached the end of the column, the power is turned off, the gel is removed, and staining procedures can be done to observe the zones or the bands of the proteins. So it's just like the thin layer chromatography that we discussed earlier in this case. For the horizontal page, we have the gel held horizontally. The setup is almost the same, except that the slab is used and is held horizontally. So this is the most common type nowadays. The advantage it offers is that you can have different samples applied to the slab. So these are different samples applied to the slab on the same slab. So it's easy to compare because the separation is taking place under similar conditions. So for visualization, staining is performed to allow for visualization of the components. So you can see that the samples are applied on the cathode end and they migrate towards the anode. So one of the challenges that I encountered with gel electrophoresis is overheating. So remember, we are applying a high voltage across a resistant medium. So overheating or temperature can increase, and this increase in temperature can denature proteins. And because of that increase in temperature, then solvent may evaporate, in which case the migration through the slab may be affected because of the different hydration conditions of the slab. Convection currents, this is as a result of the differences in the medium, and the current may flow and result in mixing of separated zones. So this would affect the ability of the slab or the separation to occur, or the efficiency of separation will be reduced by these convection currents. Any questions? Okay, if there is no question, then we move on to moving boundary electrophoresis. Capillary electrophoresis has a high resolving power, and it's also a very fast process. It can accommodate different types of samples and can be easily automated. So it's very similar to HPLC that we discussed already. It is also a sensitive technique and can be used to analyze very small samples. The components of the capillary electrophoresis there is a source and destination vials. So we have, this is the source vial and the destination vial. So these vials contain an electrolyte solution or a buffer, and these two are connected to the power supply. So we have a high voltage power supply. This voltage is about between 5 and 30 kilovolts. The connection is through platinum electrodes. Then we have the column or the capillary, whose heads are dipped into the source and destination vials. The capillaries that are used are usually flexible, and they may be bare or coated or polyimide coated fused silica capillaries. So they may be bare, just fused silica, or they may be coated with polyimide. They have an internal diameter of between 50 and 100 microns, and the length is between 25 and 100 centimeters. The capillaries may be just full of solution, or they may contain some chromatographic matrix. So they may be just hollow, or they may contain chromatographic matrix. If separation by molecular sieving is desired, the tube may be filled, that is capillary tube, may be filled with polyacrylamide or SDS polyacrylamide. So that would help or would achieve, would be like doing gel electrophoresis, but using a capillary tube. The capillary also has a transparent window near the cathodic head. So that would be this point. And this allows for attachment of a detector, usually a UV visible detector, just like in HPLC. So you're able to monitor whatever is coming out of the capillary tube real time. So various detectors may be attached to facilitate that detection. And similar to HPLC, it is possible to get a plot of detector response against time. So you're able to get an electropherogram, which is equivalent to the chromatogram. If a photomultiplier detector is attached at that point, then you are able to get a construction of mass to charge ratio of the ionic species. So the rest of the components are data handling system or integrator. So these are able to give you the area under the curve, and therefore you can relate that to quantities. And you may also have a temperature control device, so something to control the temperature of the capillary tube, and therefore get better reproducibility during the separation. So the basis of separation in capillary chromatography, the high voltages that are used can generate two types of movements. And these movements will be different for different ions or for different solids in the mixture. The first type of movement is known as electroosmotic flow. You can relate that to osmosis, but under the influence of electric charge. Electroosmotic flow occurs as a result of bulk flow of ions. So in this column, you can see that the sample is being applied at the anodic head. You compare that with the slab where the sample was applied to the cathodic end. This one is being applied at the anodic head. When the column used is a fused silica column, there are free Silano groups on the surface, on the internal surface of the columns. When the pH of the buffer is above 4, the Silano groups are ionized. You recall, we said the surface is going to have Silano groups, and depending on pH, these groups will be ionized, releasing hydrogen ions, and negatively, so the surface of the capillary is going to be negatively charged. So these positive hydrogen ions and other positively charged ions in the medium sort of form a double layer of charge on the surface. And when the high voltage is applied, these positive charges are going to be pushed towards the cathode. And due to the large number of the positive charges, they will carry with them neutral and negatively charged ions as the flow of the buffer solution moves towards the cathode. So it's like we're pushing something, and because of the attachment to the other parts, okay, there is bulk flow of buffer which sweeps everything towards the anode. So this is what is called electroosmotic flow. So this force is not as, or this flow is not as strong as electrophoretic flow. So electrophoretic flow is due to the force experienced by the ions as a result of the electric field. So the total force experienced by the ion will be a product of the electric field and the charge on the ion. So if you have two positively charged ions, one with a higher charge, the one with a higher charge will experience a greater force, and therefore would expect it to move faster if all the other factors were constant. So electrophoretic movement is due to the charge, the interaction between the charge of the ion and the electric field. So this flow is much greater, as we have mentioned, than electroosmotic flow, and therefore separation will also occur because of that difference. So peak broadening may be observed in capillary electrophoresis due to longitudinal diffusion, just like in chromatography, and electrodispersion. So remember those currents that we mentioned earlier that occur, convection currents that may occur within the column. So the applications of electrophoresis in the pharmaceutical field. Electrophoresis can be used to quantify protein products. So this is determine how much of a particular protein is present. It can also be used for purification, especially during studies where you maybe want to separate or purify a large amount of protein. Can also be used for limiting impurities. So this is ensuring that the impurities in a product or in a raw material do not exceed a certain amount as per monograph requirements. And finally, they can be used for analysis of small molecules. So not only proteins, but also smaller drug molecules. You can read more details on those applications and others, among others. Any questions on electrophoresis? Yeah. So if there are no questions, then we can briefly look at affinity chromatography. So affinity chromatography is a technique that relies on specific interactions between the analyte and the stationary phase. So these very unique or specific interactions may include interactions like between receptors and ligands, enzymes and substrates, antibodies and antigens. So those are the kind of interactions that are utilized in affinity chromatography. So the stationary phase is selectively retains analytes and can also be used to study biological interactions. So this technique can be used for separation or for study of interactions between the analyte and the stationary phase. So a high degree of purification can be attained with affinity chromatography. What are the advantages of affinity chromatography? One of them is that it has very high selectivity. So any molecule that is not interacting with the stationary phase will not be retained and therefore easily removed. And because of that high selectivity, resolution is also very high. So only molecules that can interact with the stationary phase will be retained, which may be just one type of molecule in a mixture. And then it has very high capacity. So a large amount of analyte can be purified by through a single run. So the principle of separation is just the normal key and lock that we use to explain interaction between those pairs of substances, either immunoglobulin and antigen, or antigen antibody, or enzyme and substrate, or receptor and ligand. So the key and lock, meaning that they have some complementarity between the two sets of materials. What are the types or the method types? for affinity chromatography. So if we were to discuss the proteins, we can say that affinity chromatography is broadly divided into two. So the first method uses a naturally occurring structure or amino acid sequence as the binding site. So this type of binding would use naturally occurring molecules. For example, albumin. Albumin has a site where it binds bilirubin. So you can use albumin to bind specific substances on this site. Also, antigens or antibodies can be used to bind specific antigens. So if antigens need to be separated, then specific antibodies can be used for that purpose. So that is the first type, just using naturally occurring sequence of amino acids or proteins to carry out the separation. The second type involves using specially engineered sequences. So if a protein is produced by artificial methods, then a unique sequence of amino acid can be incorporated. This unique amino acid sequence, which is not a normal sequence in a natural protein, would be called a tag. Okay? So this tag can be used now to bind to the specific binder on the stationary phase. So these tags are available commercially, and therefore it is a convenient method for separating proteins. The most common tags which are used are polyhistidine tags, and glutathione S-transferase. So those are the two types of tags that have been used commonly. So which is the support material that you find in affinity chromatography. Just like the other method that we just discussed, you can have carbohydrate based or synthetic based. So the carbohydrate is agarose or cellulose, and this type of support is only applicable for low pressure operations. For synthetic, we can have silica particles, monolithic materials, or synthetic polymers. So this can be similar to those found or used in HPLC. And when they are similar to those used in HPLC, they can accommodate, or they can become operational even at high pressures. And therefore, affinity chromatography can be combined with normal HPLC to achieve specific goals. So what factors need to be controlled for efficient affinity chromatography? Yes, Walter?
When you say that affinity chromatography can combine with normal HPLC, do you mean normal phase HPLC or reverse phase HPLC?
Either. Okay, it can be combined with either, but if you use normal reverse phase, you are likely to get denatured protein. So it depends on the goal of separation. If you want a functional protein, then it will determine the type of chromatography you can combine with them. Is that okay?
Yes.
Okay. So what factors need to be controlled in affinity chromatography? The first is the buffer. Buffer is used to control the pH, or the type of buffer that is commonly used is the phosphate buffered saline. So this phosphate is the normal, it's more common in biological systems. So it will not generally affect the behavior of proteins. The other consideration is elution, because this affinity chromatography is done in 2 steps. The first step is adding the sample, and the binding takes place. But then you may need to get that bound material free, which is the elution step. So generally, this can be achieved by using a buffer with a low pH. Okay, with a low pH, but this risks denaturing the protein. So if you are to look at the 2-step process of affinity chromatography, the first step is the application of the sample and the binding, and then there is a washing step. So washing, you're simply removing any material which is not bound, and then the elution step, where you use a different buffer so that whatever was bound is released. And you can see that in that step, you can regenerate the column for further use. So if you were to have a chromatogram, you'd get a chromatogram like the one attached there. So the first peak will obviously contain any unbound analyte, and to be a mixture of anything that was not bound, even the analyte that was excess, okay, that was more than the capacity of the column, will be present in that first peak. The second peak will contain whatever had been bound to the column. So you'd get only 2 peaks, and the 2 regions free of any sample are where the change in buffer is being carried out. Okay, so in affinity chromatography, you will not get more than 2 peaks like in other types of chromatography. So isocratic elution can also be carried out, a variation to the above method, and affinity extraction can also be done. So those are the different variations of carrying out affinity chromatography. You can get more details on those for your knowledge. Any questions? Okay, so if there are no questions, I'd like to end there. Tomorrow we can have a physical class, and then we'll see how far we can go on chromatography applications. The details of how to apply the information gained from a chromatogram for real life use. So we end it there. We meet tomorrow. Have a good morning and a good afternoon.